The next recipient this year is Levi Garraway. And to introduce him and his body of work, I asked the physician-in-chief, Jose Baselga, to come up and do the introduction of Levi. Jose.
Thank you, Greg. It is a great pleasure to introduce Dr. Levi Garraway as the recipient of one of the Paul Marks prizes in cancer research this year. We all know Levi work well. He is a worldwide leader in cancer genomics, and his work has had a particular impact on the genetic understanding of a number of tumors, in particular melanoma and prostate cancer. Levi also has made a number of contributions in the study of resistance to targeted therapies, and has been a major innovator and one of the key drivers of the field of personalized and precision-based medicine. Levi obtained his undergrad degree in biochemical sciences from Harvard in 1990, and then both an MD and a PhD degree from Harvard Medical School in 1999. Then he went on to be an intern and resident in internal medicine and a chief medical resident at Mass General, and then he joined the Dana-Farber Cancer Institute as a fellow in medical oncology, and he spent a number of years in the lab of Bill Sellers until he became independent in 2005. Levi is currently an associate professor of medicine at Harvard Medical School. He is a senior associate member of the Broad Institute of Harvard and the MIT, and he is the co-leader of the cancer genetics program at Dana-Farber Harvard Cancer Center. And as I mentioned, he has multiple contributions, and he needs to talk about them. I don't want to mention them in detail, but just to enumerate them, he identified what is being considered as the first lineage survival oncogene, MITF, in melanoma. And since this initial discovery, other lineage survival genes have been oncogenes have been identified in other tumor types, including lung cancer. He also identified that over 70% of melanomas harbor a recurrent base mutations in the promoter region of the telomerase gene. So there's a little of connection here in this field. And he has led and published, together with Mike Berger, that was in his lab, and he was the first author on this beautiful publication in Nature, the first whole genome study sequencing study of prostate cancer. He also has worked at identifying mechanisms of resistance to a number of therapies, including BRAF inhibitors and MEK inhibitors, and studied multiple new oncogenic signaling pathways in melanoma. And I'd like to focus just at the end in two aspects of his work that I think are extremely important for those of us who are trying to develop the field of cancer therapeutics. He has been the senior leader on the cancer cell line encyclopedia effort that was published in Nature last year that led to the molecular and pharmacological annotation of over 1,000 cancer cell lines. This is a very powerful tool to identify therapies that are specific to well-defined molecular profiles. And then finally, he has been one of the real leaders in identifying actionable mutations in tumors and in developing a massive parallel sequencing efforts in tumor samples that are usable, friendly to clinicians, and that can help identify mutations that we can then design therapies against them. He's a founder of Foundation Medicine, a company that is enabled this test being applied across many centers in the world. So tremendous contributions to date. And again, he was telling me during lunch that he thinks that the best is yet to come. So, and I'm sure that he's going to be extremely productive in years to come. And he's working in how to devise combination therapies that are synthetically lethal. And I'm sure that that will happen. So Levi is a prodigious scientific talent, a force of nature, but he's more than that. I think Levi is the full package, and he is, in addition to being such a brilliant scientist, he's also a superb clinician. He's a fantastic colleague and collaborator, and he's a superb mentor. So please join me in welcoming Dr. Levi Garraway to the podium. Thank you.
Well, thank you very much, Jose, for that wonderful introduction. And I, like Simon, I too have several thanks to make. I'll begin the thanks by thanking my family, many of whom are here today. Actually, one important member of my family who is not here because he passed away of cancer in 1999 is my father. But if it wasn't for that, ironically, I would probably not have gone into cancer research, but certainly want to note that. But my mom is here with her husband, Ira, as well as my two sisters, Isla and Doris. My son is here somewhere, my daughter and their cousin, and particularly importantly, my wife, Giselle, who puts up with me for many nights and weekends when I'm either in the office working or traveling somewhere, giving a talk or writing grants or what have you. So very grateful for my family. I'm also grateful for many professional mentors, particularly several whom actually have won this prize before, people like Bill Kalin and Todd Golub and Matthew Meyerson at the Dana-Farber, and certainly my laboratory mentors over the years, starting from graduate school with someone named Steve Beverly, who worked in genetics of a parasite but taught me how to think about genetics, and then Bill Sellers, as Jose mentioned, who taught me how to think about cancer genetics. So very grateful to the members of the committee for selecting me for this prize. Very grateful for Dr. Marks and the opportunity to meet him and the inspiration that this prize means to help us continue working, because obviously we have a lot more work to do. People are still dying of cancer in our institutions. Until we make that problem go away, we can't be too proud, but very grateful for the honor today. So I'd like to spend the next few minutes giving you a high-level summary of much of the work that we've been doing and how we think it's going to help move cancer forward. And to begin, I'll just point out that this notion, actually, I think for those of you who've been paying attention to Simon's talk, you could, of course, anticipate that because of problems of DNA repair, when they go wrong, that leads to cancer. So it follows, therefore, that cancer is a disease of the genome. But this idea that cancer is a genomic disease is not new. In fact, it's at least a century old. It was, in the scientific literature, it was first postulated by Theodore Bovary, a pathologist who was visualizing chromosomes under the microscope and looked at normal cells and cancer cells and postulated that chromosomal defects might lead to abnormal cell proliferation. And this is sort of a schematic that was from one of the early papers. And so this idea has been percolating for a century, but it took 6 decades at least for the notion to really be cemented as part of our fundamental understanding. And of course, that notion came from seminal experiments from the labs of Harold Varmus, who was president of this fine institution for many years, together with Mike Bishop and several colleagues, who found, looking, of course, at the model of the Rouse sarcoma virus and its ability to transform chicken cells, that it turned out that, lo and behold, the transforming property, or SARC, was not some viral sequence, but rather it was a perverted variant of our own genome. It was a mutated variant of our own genome. So that really gave birth in the modern era to the genome theory of cancer. And of course, we've now seen the next 40 years have multiple revolutions, not only in experimental approaches, but increasingly in technology and analytical approaches that have allowed us to understand with great precision the range of abnormalities that go wrong, but also in some ways how complex and challenging our problem still is. So for this talk, I'm going to have 3 sections. One is how our work has helped to highlight new insights into biology that have come from cancer genome analysis, which I'll start with, but then I'll move into how that has brought us to think about the concept of tumor dependency, how we can be smarter to target tumors based on particular events, and how this notion is moving its way into the clinic more and more every day. So in terms of new biological insights, certainly I think there's little argument that the explosion of cancer genome knowledge that has come from massively parallel sequencing technology has really peppered our entire sort of fundamental landscape of the cancer cell and the basic processes that are perturbed to give rise to cancer. So we knew even before we got into large-scale sequencing studies that signaling pathways such as the MAP kinase pathway and the PI3 kinase pathway and receptor tyrosine kinases that sit at the membrane and direct those pathways were deranged, often by mutation, in many cancers. But what the aggregate knowledge of cancer genomics over the past 6 or 8 years has taught us is just how vast the perturbation is, extending far beyond the signaling network. So for example, we've learned through studies from the Johns Hopkins group that metabolic genes such as isocytrate dehydrogenase genes are mutated, and that can be a major driver of cancer. This is something that we certainly knew that there were abnormalities in metabolism, but we didn't really understand how the genetics might give rise to that. And then one of the major discoveries is the aggregate knowledge of the number of chromatin modifying enzymes and epigenetic enzymes that are mutated in cancer. And this, I would say, collectively has been one of the most profound discoveries, that is how chromatin biology and epigenetics is deranged across so many different cancers and the mutations of the genes they're in and how they differ in particular lineages. Now, of course, that gives rise to a transcriptional program, but now in the past 2 or 3 years, there have been a variety of discoveries about splicing factors that are mutated. This is something that we hadn't expected at all. This came completely out of left field until about 2 or 3 years ago. And then recent studies of translational proteins and protein homeostatic ubiquitin machinery mechanisms. So in essence, there is a vast array of fundamental cellular processes that are in aggregate mutated to give rise to cell states that lead to the cell autonomous hallmarks of cancer. Now, to say this in generality is really not to tell you anything, because the skeptic could say, well, we always knew that all of these processes must be important in cancer, but what cancer genomics has done is has had a centering effect in the particulars that go wrong and where we need to focus our attention in specific cancers. So one example of this is a study of the genomes, and actually in this particular case it's the exomes, or the protein coding subset of the genome, in melanoma. And this is how we tend to view the results of these exome studies. So this is you can get sort of a bird's eye view of the landscape of mutated genes. And so what you get here is a list of genes, some of which we knew about before. So we've known for a long time that the MAP kinase pathway genes, BRAF and NRAS, are highly commonly mutated in melanoma. We've known about mutations in other signaling effectors or regulators such as PTEN in the PI3 kinase pathway, the INK4 locus, P16 for the CDK pathway, and then copy number alterations such as MITF, which is a lineage survival oncogene that we discovered several years ago, as Jose mentioned. So we knew about a lot of these, although the prevalence maybe needed to be defined. But it's shown in bold are a variety of genes that tell us about pathways that we didn't know about in melanoma, but can have served in many cases as focal points for discrete understanding. So for example, RAC1, which is a RAS-like GTPase mutated in a subset of melanomas, and then there are other RAC family members that are also mutated at lower frequency. This is a protein phosphatase, which points to a pathway that may regulate cell division that we hadn't appreciated at all. There are genes like ARID2 and EZH2. These are, again, chromatin modifying enzymes or chromatin compaction regulatory mechanisms that we didn't know about in melanoma, but now they're focal points of specific studies. And then FBXW7, which is a member of a ubiquitin ligase, so the protein homeostatic machinery. So in melanoma, we now have particular focal points for several of those processes that have prompted a variety of hypothesis-driven studies. And so this is exactly what we would hope to get from these types of systematic studies that massively parallel sequencing has enabled. But arguably, some of the most unexpected discoveries don't fall necessarily just within the coding sequence of genes such as these, although those are certainly important. They fall in what we in the field have colloquially come to call the dark matter of the genome. So we use this term, which of course is maybe not the physicists might get mad because it's sort of not the right way to think about dark matter, but nonetheless, we consider it, these are the regions of the genome that we cannot easily interpret. So regions in between the genes, regulatory regions, intergenic repeat-rich DNA, and in some cases you can think of the non-focal copy number alterations as falling into this. But certainly the fact that we can sequence, and we are sequencing whole genomes, allows us to at least ask the question of whether or not there's mutational activity in the intergenic regions, the regions that we don't often think about that might be relevant for cancer. And so work by Mike Berger, who's now here at Memorial Sloan Kettering, enabled us to sequence a couple of dozen whole melanoma genomes. And then Iran Hodas, who is a computational biologist in the lab, asked the very simple question is, are there any nucleotides in the melanoma genome that are mutated more than you would expect by chance? And truth be told, even twice would be more than you might expect by chance when you're talking about 3 billion bases. But we were shocked to discover that there were 2 nucleotides that were mutated highly recurrently in more than 70% of cases that we ultimately studied of melanoma. And these 2 mutations, these 2 nucleotides, were of particular interest because, as Jose mentioned, they resided upstream of the promoter region for the telomerase reverse transcriptase gene, the TERT gene, and you've just heard a lot about telomerase and its importance in genome integrity, and it turns out that dysregulation of telomerase is a driver of most, if not all, cancers. So obviously these seem to be important from the standpoint of where they were situated, but what was even more interesting is that both of these mutations ended up generating a consensus 11 nucleotide stretch, at the center of which was a binding site for a category of transcription factors called ETS factors. And we knew that ETS factors were critical in melanoma because, like MITF, they cooperate, they actually function downstream of the MAP kinase pathway to drive the oncogenic transcriptional program. So it was particularly appealing to postulate that perhaps these would be activating events, and maybe even ETS factors would be among the factors that would activate them. And so the sort of initial but pertinent experiment that nailed this was a reporter assay, which essentially cloned these various promoter mutations upstream of luciferase and showed in a variety of contexts that there was consistently upregulation conferred by either mutation. And it's not massive upregulation, it's sort of in the 2- to 4-fold range, but you can imagine, we know that the activity and expression of telomerase is tightly regulated during cell division. So even a bit of dysregulation over the course of tumorigenesis could be sufficient. And in fact, this was of interest because it hadn't really been all that well understood how telomerase becomes dysregulated in cancer. We knew that it is, but we didn't necessarily know exactly how it might be. And so this may be one important mechanism by which this would happen. And so now others have gone on. This is an example of a study that was done collaboratively, including the group at Johns Hopkins, to look at these particular mutations across a whole variety of cancers. And you can see now that there are multiple tumor types where these mutations are incredibly common, including brain tumors, primary glioblastoma, but also bladder cancer. More than half of bladder cancers have these mutations. Liver cancer, about 40%, and then other types of tumors at lower frequency. So we often talk about RAS, the classical oncogene RAS, and codon 12 of RAS being perhaps the most mutated codon in all of cancer. Well, you can now make a case that these particular nucleotides may be the most commonly mutated bases in all of cancer. So it's quite striking that those bases would be landing not within a coding sequence of a gene, but in the promoter of a gene. So that was one unexpected observation that falls outside of the genes per se. And another one came from studies that we did, and actually several of these studies were also led by Mike Berger, so you guys at Memorial were lucky to snarf him away from us. But these studies came in prostate cancer. So one of the observations that came out of sequencing the entire genome of prostate cancer was that there was a category of rearrangements that occur in those genomes in particular that we had never seen before. And so this is how we view our whole genomes. We call these circus plots. So you put the genome, all the chromosomes in a wheel, and then the spokes in the wheel are rearrangements, pieces of one chromosome that are broken off and attached to the other. And what's being depicted here, each color is encoding a particular set of complex chains of rearrangement that have occurred that span multiple chromosomal loci across, in fact, many independent chromosomes. And I don't have time to go into great detail about how we think these arise, but the essence of it is that we postulate that these disparate regions of the genome become physically co-localized at some point during the life of the cell, and one prominent candidate for what may co-localize these is transcription. So it turns out that transcription will often occur in the concept is hubs or physical locales where the transcription factors reside, and so disparate regions of the genome can come to those hubs. But what could happen is if a break such as the type that Simon studies, or maybe a different type, were to happen, and then there were to be propagation of breaks for whatever reason. We don't really understand what would initiate and propagate these, but you can imagine, though, that if this were to happen, and there was breakage followed by misrepair of the disparate regions that were physically co-localized, you would get exactly the pattern that we see in these chains. And so we called these chains, we felt like they were so prevalent and that they needed a name, so we called them chromoplexy, which means chromo, of course, is chromosome, plexy is to weave or braid. And what was shown on the last slide was that almost all prostate cancers have at least one of these chains, and 2/3 of prostate cancers have 2 or more chains. So these are not only are they prevalent, but they are happening presumably in a punctuated fashion over the course of carcinogenesis. And in fact, we were able to show in some cases that these chains, these events, these discrete events, appear to be subclonal in nature. Now, the other important component is that they commonly dysregulate cancer genes. So here's an example, a schematic of one of these events that is pounding the locus of P10, which is a tumor suppressor that regulates the PI3 kinase pathway. And indeed, if you consider, for example, the ETS transcription factor rearrangements that are highly prevalent in prostate cancer, the majority of them have arisen as a result of a chromoplexy type event. So this is clearly an event that's not just happening as a fascinoma, but it is apparently a means of dysregulating prostate cancer genes, which means that it is important in the evolution of prostate cancer. And so this recognition made us realize that if you think more broadly now about the models of tumor evolution, historically we have viewed tumor evolution as a neo-Darwinistic fashion where every cell division gives you some mutations, and you collect mutations in the progeny of cells over the course of one's lifetime, and at some point you cross some threshold where that gives you cancer. And of course this is obviously a very important mechanism of cancer, but recent studies have made us realize that this is not the only way to think about cancer, and one example is the discovery by Phil Stevens of the Sanger Institute of this phenomenon called chromothripsis, which you can think of as a catastrophic event that can potentially boost cells in one fell swoop over the hump towards cancer. But we think chromoplexy may lie in the middle, sort of akin to the punctuated equilibrium model of species evolution that Stephen Jay Gould pioneered, wherein these chromoplexy events may occur overlaid across a neo-Darwinistic pattern. And so punctuated evolution in prostate cancer and perhaps in other types of cancers as well, it could be a critical mechanism through which this particular malignancy arises. And this is important in the sense that if we had not done whole genome sequencing and looked for these types of aberrations, we would not have been able to appreciate this potential model. And now, of course, this sets up a whole series of studies of why do these events form. Could one dial down their prevalence and their occurrence, and would that be a way of preventing prostate cancer at some point in the future? So the next sort of portion I'd like to briefly touch on is how these biological insights inform tumor dependency and resistance. And so really, of course, where we're trying to get is shown here at the bottom left, which is cure or long-term control. And the genome studies have really have been driven by the idea that if we understand recurrent alterations, we will identify cellular pathways or processes that give rise to dependencies that are druggable. And so the presence of rational therapeutics and when you can match these properly, has led to substantial responses. However, we're not here most of the time because resistance arises. And so we need to understand both the spectrum of dependencies and the mechanisms of resistance so that we can think about what combinations need to be provided. Now, in melanoma, as I've already mentioned, the MAP kinase pathway, which is schematized here, is commonly dysregulated. So activating mutations in this gene BRAF is one of the most common ways of doing this, and there are, as you all know, inhibitors, targeted agents that work extremely well in the clinic that target both RAF and its downstream effector kinases MEK and ERK. And these, of course, have led to remarkable clinical activity, as you can see by the Kaplan-Meier curve. And actually, what we're looking at here is work that builds on pioneering efforts, some of which was done here at Memorial Sloan Kettering, where a RAF inhibitor monotherapy does a lot better than historical controls, but the combination extends you even farther. And these waterfall plots show you the magnitude of clinical benefits. If you're going below the line, you have clinical benefits. So almost all patients are having clinical benefit. But there's a lot of resistance here. There's a lot of intrinsic and acquired resistance to these inhibitors. So the intrinsic resistance can be appreciated by the fact that if you use an objective measure of tumor response, you find that a substantial proportion of patients don't even achieve this objective measure, even while they're benefiting from the cancer. And of course, we're still measuring tumor progression in months as opposed to many years. So we need to do a lot better here. And the basis for doing better could in part be understanding mechanisms of resistance. Now, we, of course, and others have thought about applying these same genomic tools to studying resistance. But here the concept is to take biopsy material of patients prior to treatment and following relapse using one of these targeted agents, sequence the genomes and understand using both analytical and biological knowledge what the resistance effectors might be. So now this is a landscape view akin to what I showed you before, but this is now the resistance landscape. So it looks somewhat different than what I showed you before, but some of the players are the same. So you can bend these into a couple of different categories. These genes here are MAP kinase genes or MAP kinase pathway regulatory genes, which makes perfect sense because the idea is if you block MAP kinase signaling, but you can turn it back on at some other point downstream, you'll get resistance. And so this turns out to be a prevalent way. So what we're showing here is mutations at the point of relapse. Now, there's another pathway, the PI3 kinase pathway, which I've been talking about, which also appears to undergo resistance-associated mutations. These may not drive proliferation, but they may enable survival of the cells. And then there's a variety of other genes that occur at less frequency that have provided us new insight. Now, this is an exciting landscape in some ways because it's actually very different than the steady state landscape of melanoma. And in fact, what I think this tells us is that although a lot of our cancer genome studies have been focused on primary tumors, we've done almost nothing looking at the landscape, mutational landscape of tumors that arise after effective treatment. And of course, it's patients who relapse after treatment that end up, of course, being the medical challenge, and often really we run out of options there. So we certainly need to look at this better. But a closer look at this also is a bit sobering, and it's for a variety of reasons, and I'm going to summarize those reasons on the next slide. So here again is our MAP kinase signaling pathway. Many of the mutations associated with resistance will reactivate MEK and ERK signaling. But we also know by looking at the landscape and its dynamics that there is a large amount of heterogeneity, not just in terms of individual resistance mechanisms, but we can often see multiple independent validated resistance mechanisms arising in the same tumor. So you can't just sequence a tumor and say, oh, here's my favorite gene. There often are more than one of those genes. And so this is one challenge. The other is that you could probably appreciate from the landscape that there is what we call a long tail. There's not a few highly prevalent mechanisms. There are many mechanisms. And then what this tells us is that we may be, even with our most state-of-the-art approaches, undersampling the spectrum of resistance, which makes it concerning that although we know that we need to attack this locus and we know that we need to overcome resistance, the problem could be akin to something like this, where it would be futile to just pick one or another of these resistance mechanisms. We need to be smarter. And so the question is, can we hit the resistance mechanisms at any point of coalescence at the bottom of this? And so the way that we have gained insights into this is by moving from sequencing to kind of systematic functional studies where we've been taking each individual gene, or in this particular case about 15,000 open reading frames, expressing them in drug-sensitive cells and looking for individual genes that can confer resistance. And so we've done this using drugs all the way down the MAP kinase pathways, RAF, MEK, ERK inhibitors, and the combination here. And we've mapped out the genes that are sufficient, at least in an experimental system, to confer resistance. And right away, what becomes prevalent is that there are multiple transcription factors that are capable of conferring resistance. And if we take this information together with a variety of other genes that have come out of these studies and been validated across multiple contexts, we can appreciate that there are bypass modules that can be activated at multiple points. So there are multiple resistance genes that were identified that point to a module in particular that then feed into a transcriptional output. And one of the prominent members of the transcriptional output is MITF, the same lineage survival oncogene that we discovered a while ago. But what was appealing about this observation is that this module is, which we're showing here in BRAF mutant melanoma that's been treated with the RAF inhibitor, is highly reminiscent of a known signaling mechanism that's present in non-transformed melanocytes. The pigment-producing cells of the body have this mechanism which feeds into MITF and makes them melanocytes naturally. And in fact, when the BRAF mutation occurs, what happens is that this signal is now usurped by the MAP kinase pathway. This module becomes dampened. So it actually makes perfect sense that if you were to reactivate this module, this would be a resistance mechanism. And we now have initial clinical evidence, which I don't have time to get into, to show that this appears to be going on. But the reason why I brought this up in the context of dependency is that this may give us an opportunity to attack a point of coalescence at the bottom. And the reason for this is that David Fisher several years ago showed that MITF could be, the levels of this protein could be downregulated by using histone deacetylase inhibitors. So these are inhibitors that are attacking the chromatin machinery. So these are already in the clinic. In fact, there are HDAC inhibitors that have been approved for certain cancers. So the idea was perhaps if you combined an HDAC inhibitor with the RAF or MEK inhibitors that are already in use, could this be a way to overcome resistance? And some preliminary data from cell culture models has suggested that this might be the case. I won't go through the details, but we're looking at growth of this particular resistance mechanism in the presence of either MAP kinase pathway inhibition by itself or the addition of one or another HDAC inhibitors. And the take-home message is that when you add the HDAC inhibitors to the RAF or MEK inhibitors, you can suppress this particular resistance mechanism. And so now we're in active conversation with pharmaceutical companies about potentially doing a trial with this type of combination. And so what we're thinking is, of course, when you think about using genetics to point to therapeutics, of course, stacking the deck against the index mutated pathway, considering parallel pathways such as the PI3 kinase pathway. But now, can we think about attacking the transcriptional output at the level of chromatin biology? And could this be a mechanism to think about combinations going forward? We think the answer is yes. So in the last 2 minutes, I'm just going to briefly summarize how these types of insights of biology and dependency are influencing precision medicine and cancer. And so for the concept, of course, has been that when we biopsy a patient and diagnose or gain some understanding about the nature of the prognosis of the cancer, we want not just to look under the microscope to make the diagnosis of cancer, we want to get a readout, a genetic or molecular readout that might tell us about what kind of clinical action might be feasible. And so over the years, we've piloted this idea, starting with an earlier genomic platform, mass spec genotyping. We, several years ago, published the concept that if we configured mass spec genotyping into a mutational profiling mechanism, this could be a rapid way of reading out the salient information in a clinically actionable way. Sorry for the, so now what has happened is OncoMap migrated through development at a center at the Dana-Farber and then finally was rolled out clinically at the Dana-Farber and Brigham and Women's Hospital, and many other cancer centers have taken similar approaches and rolled this through. Now, what we did when massively parallel sequencing came along was reason that we could again take the same principle, starting not just with the entire genome, but focusing on a subset of cancer genes that are important. This development has also happened not only at the Brigham and Women's Hospital, but now many cancer centers, including Memorial Sloan Kettering and others, and commercial avenues such as Foundation Medicine are using this same kind of concept to carry out mutational profiling to guide clinical trials. Now, of course, the technology doesn't stand still, so now the question will be, well, what if you brought even a more state-of-the-art, a more comprehensive approach forward, such as whole exome sequencing? But certainly there are other technologies emerging. You know, can we continue to determine where and how and if this kind of information will be transformative in terms of guiding treatment. But certainly what this is doing is fitting into a new framework for how we think about clinical research and translational research in cancer, where it very much is now not just in the laboratory, but in partnership with our patients who are struggling with disease. And so the idea is that patients, when they enter our cancer centers, we are thinking about sampling their tissue not just once, but at multiple points over the course of the disease. And the profiling and the interpretation will guide the choice initially of clinical trial enrollment, but eventually of clinical decision making. But not even at the point of initial treatment, but also to determine whether or not the treatment is working. So we need to be able to follow whether the drugs are suppressing the pathway in the right way. And then if the patient were to relapse, we need to do this once again to understand how did the landscape change? What does that tell us about salvage therapies that should be tried then, or perhaps new therapeutic combinations that be given to the next patient? So it's a daunting time because getting this logistically to work is challenging, and yet it's an incredibly exciting time because we know that the knowledge of the rational basis of cancer, starting with the genome and other molecular tools, are going to be essential for our ability to bring the smartest therapies to our patients. And as Jose said, I do believe that the best years are yet to come because we have yet to put this in place systematically. But once we are doing this, I would predict that many more patients are going to benefit in a lasting and durable way from the drugs that we are working so hard to bring to them. So in summary, I hope that this talk has told you about how systematic studies of the genome have given some perhaps expected but many unexpected insights into biology, how those insights are guiding our ability to develop combinations based on not only the dependencies but the resistance mechanisms that we know about, and finally how all of this is coming together with increasing acceleration to drive precision medicine. So I will stop there. There are, of course, many people who have been involved in this work, and several of them are highlighted on this slide, but I'll take time for questions if there's time.
Questions?
Very nice rationale for the use of HDAC inhibitors together with the inhibitors of pathway. I was wondering if you think, or if you have data about the combination of about a rationale for using HDAC inhibitor together with PI3K, meaning if the same results can apply also for inhibitors of the PI3K pathway.
Yeah, so it's a great question. So the question is, we proposed the idea of combining MAP kinase pathway inhibitors with HDAC inhibitors, and so the question is, could there be a rationale for doing the same type of combination but using PI3 kinase inhibitors instead of MAP kinase? And the short answer is we haven't specifically looked at that. The reason why we went to HDAC inhibitors was because we knew that they had a specific effect on MITF, which is one of the key transcriptional effectors downstream of which can now be involved in resistance. So it was a mechanism-based or hypothesis-driven choice. So I think to assess that, we would need to ask ourselves, what are the mechanisms of resistance, and are there similar transcriptional output issues downstream of the PI3 kinase pathway? Are any of those are down modulatable by an HDAC inhibitor? And actually, we are doing that right now. In fact, we have just completed an ORF screen of a PI3 kinase pathway inhibitor, and the data is brand new, so we're literally just staring at it. But so I think we will have the opportunity to address whether it's an HDAC or some other kind of chromatin inhibitor that might be relevant. It's a great question.
Other questions? Yes.
So that's a great question. So one of the problems with these landscape studies is that there are a lot of genes. There are a lot of statistically significantly mutated genes, but many of them are mutated at lower frequencies than are sufficient to draw rigorous conclusions about mutual exclusivity. So we call this the long tail, which is there are a few very commonly mutated genes and then a long tail of genes that are in the 2 to 3% mutation frequency. So to actually answer that question, we would need many thousands of tumors. Now, when I say this, some cynics say, oh, you're just saying that because you want the genome sequencers to stay in business. But it actually happens to be true. If you really wanted to answer that question, you would need to sequence many thousands of tumors. Now, what I think is going to happen, because we recognize the clinical relevance of sequencing, is that that will probably happen naturally as people are beginning to sequence for clinical decision making. So I think over the years that the number of sequenced tumors will grow, and we can actually determine mutual exclusivity, which I think is a very important question. But unfortunately, when you only do 100 or 200 or 300 tumors with the long tail, there are no statistics to address it.
One last question.
In the first part of your talk, you told us about two exciting new hotspots you had found by the mutations or C2T transitions.
Correct.
And you tentatively described that to ultraviolet light. It's a very common mutation, and you find it in colon cancer. And for UV-induced mutations, you might expect transversions rather than transitions. So I would turn your argument around and say that your data indicate that melanoma is not caused by the direct radiation.
Well, so it's a very good point. So I think one of the things, so the point is the mutations are C to T transitions, which are in melanoma one of the signatures of UV damage, but we see these same mutations in a lot of other cancers where the sun doesn't shine. You know, it doesn't shine in the liver and the bladder. So how do we reconcile this? And it's a very good point. So C to T transitions are not just caused by UV. They're actually caused by, they happen in every genome that's been sequenced, you see lots of C to T transitions. So I think we got excited. The reason why we highlighted that in melanoma is because we know in melanoma that the preponderance of mutations that we see, somatic mutations, are C to T transitions, and they are not just C to T, but they have the signature of UV damage. So in melanoma, that's the case. And the reason why we took note of that is because at first, we didn't believe this result. It was so prevalent that we thought, is this really real or is it a bug? And the fact that it was a C to T transition, at least thought, well, and we were looking at melanomas, okay, well, at least it's plausible because we know this. But you're absolutely right. It would not be sensible to say in all contexts that this is ascribed to UV damage. There are clearly other ways to generate the same event.